Hi everyone. In this video, we want to talk about genetic recombination, coupling, and repulsion. So, according to the principle of segregation, alleles separate during meiosis, and according to the law of independent assortment, alleles at one locus sort independently from alleles at another locus. This leads us to what we know as genetic recombination, where alleles sort into new arrangements. In the next few slides, we will solve related problems and answer a few questions. But before we proceed, let's define some terms. What is genetic recombination? This is the process by which genetic materials is exchanged between different chromosomes or different regions within the same chromosome. We also need to know about coupling. Coupling refers to the arrangement where two dominant alleles or two recessive alleles are located on the same chromosome. Coupling orientation is also known as cis configuration. So here is what we mean by coupling. Two dominant alleles, capital A, capital B, on the same chromosome, small a and small b, the recessive alleles on another chromosome. So in repulsion, the dominant allele or one dominant allele and one recessive allele are on the same chromosome. There is capital A, small b on the same chromosome, then small a, capital B on the other chromosome. So here is the trans configuration, and here is the coupling configuration or orientation. In the next slide, we'll take a look at related problems.
Alright, so the main question that we are tackling in this particular video is, could certain beneficial cognitive traits be more likely inherited together due to coupling or separated due to repulsion? And the answer to that question is yes. Since intelligence is influenced by multiple genes, whether these genes are inherited together, which is in case of coupling, or separately, which is in case of repulsion, can impact cognitive evolution. So in case of coupling, beneficial cognitive traits such as enhanced memory, problem solving, neural efficiency are passed on together, which potentially accelerates intelligence in species like humans, dolphins, and primates. On the other hand, repulsion can slow down cognitive evolution by preventing the inheritance of full sets of advantageous traits, which leads to the variability in the intelligence within a species. So if genes for problem solving and learning speed are frequently separated, it may limit the overall intellectual progress of a species. Over time, natural selection or genetic engineering may save gene linkage patterns, which could influence the future development of intelligence in species. Later in this video, we will see an example of how coupling and repulsion leads to the cognitive evolution.
Here is the question related to coupling and repulsion. In human, two genes influence cognitive trait, memory ability and learning ability. High memory ability, big M, is dominant over low memory ability, small m, and high learning ability, big L, is dominant over low learning ability, small l. A heterozygous individual is test crossed with the homozygous recessive individual, and the observed offspring ratios are these. So for this question, we can determine whether the genes are in coupling or repulsion, calculate the recombinant frequency, and explain how this result would differ if the genes were unlinked. So when we perform a test cross between heterozygous individual and a homozygous recessive individual, we would expect to get equal proportion of each genotype. In the progeny, assuming that genes are not linked and follow Mendel's law of independent assortment. Here is a dihybrid cross between the heterozygous individual and the homozygous recessive individual. From this dihybrid cross, we can say that the genotype, they are in 1:1:1:1 ratio. But the observed frequency is far off from being in this ratio, which make us say that the genes M and L are linked. Now let's identify the parental and recombinant gametes. Since these two are the most frequent phenotypes, these are our parental gametes, and the other two having the lower frequencies are our recombinant ones. Since the parental type have both dominant alleles together, the big M and big L, and both recessive alleles together, small m and small l, we can say that these two are in coupling or cis. In our parental gametes, we can ignore the small m, small l, and small l, as we got those from the homozygous recessive parents, leaving us with big M, big L, and small m, small l. We can also write these notation as big M, small m this way or this way. Now let's calculate the recombinance frequency using the formula. The formula for recombinant frequency is total recombinant offspring by total offspring times 100. In our question, the total recombinants are 7 plus 6, which gives us 13, and the total offspring is 48 plus 39 plus 7 plus 6, which gives us 100. So the recombinant frequency is 13 by 100 times 100, which gives us 13%. So far, we have determined that the genes for memory ability and learning ability are linked because we observed more parental phenotypes than the recombinant ones. But what if the genes were unlinked? How would our results change? To understand this, we need to recall that the unlinked genes follow Mendel's law of independent assortment. This means that during meiosis, the alleles of one gene separate independently of the alleles of the other gene. In a test cross, this independent segregation produces equal proportion of all possible offspring phenotypes. In a test cross between heterozygous parents and a homozygous recessive parent, if the genes were on separate chromosome and independently assorted, we would expect 1:1:1:1 phenotypic ratio in the offspring. Each phenotype should appear in equal proportion because there would be no linkage to disrupt. But comparing this with our actual frequencies, clearly these numbers are not equal, meaning that the independent assortment is not happening. Instead, we see that the parental are much more frequent than the recombinant ones, confirming that the genes are linked and inherited together more often than the expected by chance.